All right, hi everyone. Welcome back to the Responsible Conduct of Research seminar series for our second lecture. Our topic today is data acquisition, management, sharing, and ownership. And our speaker is Dr. W.T. Godbee. He is an associate professor in the Department of Chemical and Biomolecular Engineering in the School of Science and Engineering. Dr. Godbee has been at Tulane since 2003. In 2009, he received a career award from the National Science Foundation for targeted gene therapies. The research performed with the award generated a patent for a targeted treatment for transitional cell carcinoma, and a company was formed around that technology. Dr. Godbee received his BS in mathematics from Southern Methodist University. After starting his own software design and development company in Dallas, he earned a PhD as a National Science Foundation Graduate Fellow from the Institute for Biosciences and Bioengineering at Rice University. He was a postdoc fellow at Children's Hospital in Boston and Harvard Medical School before joining Tulane. His research interests are centered on gene therapy and the manipulation of cells at the genetic level, including gene therapy for cancer treatment, controlled release applications for efficient gene delivery, and the use of gene delivery for cellular engineering. In 2014, Dr. Godbee received the Raymond V. Bailey Outstanding Teaching Award in the Department of Chemical and Biomolecular Engineering here at Tulane. In 2015, he received the Weiss Presidential Fellowship, which honors Tulane faculty with a sustained record of effective, inspiring, and distinguished teaching. And in 2016, he received the Torch Award, which is the Tulane Student Athlete Advisory Committee's award. Long before COVID nudged some activities outside of classrooms, Dr. Godbee occasionally held class outside. And here is a photo of him sketching a molecular formula during a biochemistry lecture on McAllister Place. We are truly delighted to have Dr. Godbee as our lecturer today, and I ask that you please welcome him. Dr. Godbee, will hand it over to you.
Thank you. Let's see. Lisa, is my blue title screen up?
Yes, it is.
Excellent. Okay. Thank you for the introduction. And as Lisa said, I'm going to be going over data acquisition, ownership, management, and sharing. This is the data talk. So let's begin. Oops. Oops. Let me try again. Okay. So we'll start with a quote, and then I'll boil it down for you. The institution of science involves an implicit social contract between scientists so that each can depend on the trustworthiness of the rest. The entire cognitive system of science is rooted in the moral integrity of aggregates of individual scientists. That's from the Common Sense of Science. What this boils down to is you can go to a talk, you can hear an international presentation, you can read a journal article, okay? Let's do the journal article. You read that and you see figures. Great. Those figures came from a laboratory. That laboratory had a person who collected the data to make that graph. That person who collected the data will say either sat in front of a microscope or sat in front of a machine and obtained the numbers or the images or manually assessed the data that he or she was looking at to produce the aggregate data that's represented by the graph. And that one person, the one person who obtained all these data, if that person lied, if that person was confused, if that person misrepresented what was there, then everything that stemmed from that one data point, that graph, that paper, that international talk, and maybe that international award, all of that is built on falsehood, whether it be intentional or not. So it all comes down to the moral integrity of each individual. It's all up to you to manage your data and to record it properly, because 6 months from now, you're not going to remember that day that you were looking at those cells, at least not in great detail. And so you have to document everything and be able to support your documentation if ever it gets called into question. So today I'm going to talk to you about how we obtain data, the importance of objectivity, the even greater importance of the laboratory notebook. We'll talk about different errors that you may encounter or generate while you're recording or reporting data. And then finally, who owns the data that you generate? So data acquisition. Well, this is very simply stated as the process of obtaining and recording primary experimental information. It provides a foundation, like I just talked about, for all subsequent data analysis and generalizations. And if you don't have good data collection techniques, everything that is built upon those data after the fact is tainted. The same thing with the record keeping. You may remember that these cells were exceptionally bright, but if you don't write it down at the time and you wait until that night or the next day to write down your observations that you saw in front of the microscope, you might leave out a couple of key details. And then, of course, this all goes down to proper record keeping. If not only might we publish our data, but we may generate something that's patentable, an invention, a technique, something that perhaps a company can be built around. Especially in terms of companies, we need to provide proper record keeping, because not only is the scientific community involved at that point, but also the business communities and the legal communities. So there are two types of experimentation I want to tell you about. The first is what I call the quick and dirty experiments. So you're in the lab, let's say you're in my laboratory. I have a gene therapy laboratory. One of the first things I would teach you when you come into my lab, if you don't know already, would be how to culture cells, how to make gene delivery complexes, and then to prove that you can do gene delivery, because everything in my lab is involved with gene delivery in some way. So you come into my lab and I tell you how to make the complexes. I tell you how to rinse the cells, expose the cells to the complexes, how to read the data a couple days later, or maybe the next day. Okay, and so you go in and you try. And you want to keep notes at this point because the notes are primarily to help you. You're not going to publish your first transfection experiment where maybe you made 50% of the cells turn green. That'd be a great day. And you might want to remember it on your own just for pride purposes. But later on, after we do that, I may teach you some molecular biology techniques. And by the time you get back to doing your own experiments with the genes that you constructed yourself, you might be a little fuzzy on how you did your first transfections. So these quick and dirty experiments are really there to help you, so you can go back and refer. Let's say the first time you do a transfection, you don't get such great results. So you do the experiment again the next day, okay? You will want to document what you did differently, okay? Not because it's going to go into a publication, but because as you develop as a technician or as a scientist, you want to be able to look back in your notebook and see what was different and what worked out better for you, okay? So this is why we do preliminary experiments. Not every experiment that we perform is with the intent of being published. However, your formal data must be documented, okay? When you run your experiments, you have to do every control every time. So in my laboratory, we'll do one control will be, what if we deliver this gene delivery vehicle with no gene inside, just say the polymer that we're trying to deliver to the cells. And it better come out not green. The cells had better not glow green. And after you do this enough times, you're going to know the cells will not glow green. But the one time that you skip that control is going to be the day that we have to throw your data out. Because how do you know this isn't, say, autofluorescence, which I'll come back to later in the talk. So every control, every time. And then our formal data will also include documented observations. Any observations that you had after the experiment, such as, we did get 45% transfection today, but the cells looked sickly. They had more exosomes inside. The cells they didn't have defined borders. They were swollen. They just didn't look healthy. That would be something that you want to document, okay? And eventually, you're going to want to publish a paper on your data. your notebook, your documented procedures and data collection, these are going to be what you use for your public disclosure, which is your conference talks and your publications, whether they be a paper or even a poster at a conference. So let's spend a few minutes talking about objectivity. Now, what do we consider data? Well, data are information. A datum is an information point. And we have qualitative and quantitative data. We also have what we call potential data. So quantitative is exactly what you think it is. It's the numbers, okay? So if you're in my lab and you're counting cells, you count 153 cells total, and 75 of those are glowing bright green, then you would record that by hand at your raw data. You do this several times, and you make a chart or a graph that describes the several times that you repeated the experiment. That would be an example of process data. So raw data generate process data, your charts and graphs. Your charts and graphs will demonstrate support for one point in your paper, okay? And several points in a paper generate a paper. And then a good paper will generate a talk, a conference proceeding talk. Or eventually, when you move up in the ranks, you may go talk about all the work in your laboratory. And so the paper will just be one bit that you feature in your larger scale to talk, okay? Qualitative data. That might be something like you look in a microscope and you're looking at the cells. Remember my comment earlier about the cells just look sickly. They had more exosomes inside them. They looked like they were going through apoptosis. I didn't actually run the test because this experiment wasn't about apoptosis, but it looked like they were going through. The cells are larger. They're just not happy looking cells. That would be a qualitative assessment that you make. But even qualitative data are worth something, and you should document them and treat them seriously. Another note, all the mice in group E were running around and doing backflips in the cage. That's pretty important, especially if you're testing a drug that you might eventually take into clinical trials. If it makes all the mice hyped up, you need to document that somehow. So even a silly looking comment like this is a serious comment. So maybe the dosage was too high. You would go back earlier in the notebook, see if you prepared, if anything happened during the preparation of the drug that you were giving the mice. You just want to try to explain every single observation that you make during and after your experiments. Okay, already talked about these qualitative data, pictures, slides, movies that you might make of cell division perhaps. You might make a movie of, if you're a psychologist, maybe a rat running through a maze. I don't know, I'm not a psychologist. But we also have potential data. So sometimes my laboratory, we will want to assess what's going on at the tissue level. So I do bladder cancer research. We might, at the end of the experiment, obtain and fix the bladders of the mice that were in the experiment, embed them in, say, paraffin, and then make thin sections that we then observe under the microscope. Well, that's a very lengthy process. And you don't want to go through the whole process over and over and over again. You want to process your tissue, your bladders, at one time if you can. It lowers your variability if you're observing and processing in single chunks for the entire experiment. Okay, so as these blocks with the bladders are sitting on your shelf in the laboratory, those are potential data. Those are unprocessed specimens that eventually are going to yield data for you, we hope. Of course, we all know being objective means impartial, not biased. Okay, it's easy to say these words, but I want to go into a little bit of depth talking about sometimes it's very hard to be impartial. Bias can sneak up in your experiments without you even being aware of it. We'll talk about that as well. We don't want to be motivated by personal gain when we perform our science. In fact, when we're collecting data, we should act as we're merely reporters. We run the experiment, we design the experiment as a scientist. We run the experiment as a technician, but we gather our data as a reporter. I don't want to say a mere reporter, that reporters have a hard job, but I don't want I tell my students, I don't want you to do anything other than report on the effect of the experiment. How many cells were green? How many cells died? How many cells express such and such marker? Okay, it's not up for you at this point when you're collecting data to decide if these data are good or bad. They just are. And all we are to do at that point is report them. Then we can go back and put on our scientist hat later and try to interpret the data. So also, this becomes difficult for not just graduate students, but also PIs. Avoid being personally attached to a hypothesis or a concept. That's easy to say, and if you're a beginning grad student, it's very easy to live because you don't have anything to be attached to yet. So let's say you're in the 4th year of your PhD. You're getting kind of tired. You're wanting to move on to the next stage of your career, but you just have to finish up these last few remaining experiments, okay? And the last few remaining experiments might show something you didn't predict. Well, what are you going to do? It is human nature at that point to try to make your life a little easier. And if they're ambiguous, say, well, it's still kind of consistent with everything else I've seen, and you move on. That's not good science, okay? Just like every control, every time, you need to be objective every time you collect data. In fact, why do the experiment if you're going to fudge data in the first place? You could just make it up sitting there in your chair. And that's something we would never do, I'm sure, okay? And also, if you get too attached, then you could get very defensive when you start presenting your data. And if they don't clearly support the story that you've been trying to tell the scientific community, you can get defensive, come up with reasons to explain why you saw this, reasons why you saw to explain why you saw that. That's natural. You do have to defend your ideas. It's what conferences are all about. It's what your defense for your PhDs will be all about. But sometimes if you notice something happening over and over and it doesn't agree with your story, you might need to change your story. And, pardon me, so you need to be able to step back and reassess. It's okay to change your hypothesis. We're all after the truth. We're not all out, it's not supposed to be about proving your story is correct. It's about finding the truth and reporting the truth, okay? And you have to accept responsibility for the validity of your report. So if you start fudging data and you're standing up at a national conference and somebody asks you at, sort of a grassroots level about your data, if you fudge some, are you willing to stand up there in front of everybody and tell them that you were fudging data? No, you don't want to do that. Run a clean ship at all times, and you don't have anything to worry about in that account. Personal gain and human nature. So you as grad students will, if not already, eventually you will be happy when your advisor is happy. And if the data clearly show a significance between when you give the animals your treatment and when you don't give the animals your treatment, and that significant difference supports the hypothesis that you and the advisor started out with, well, your advisor is going to tend to be happy. But if you don't present such a clear difference, the advisor might not be as enthusiastic about your work. You need to be true to the data. As important as your advisor is, as important as your advisor may be, the data and the truth of science are more important. Okay, so if you have some ambiguous conclusions, don't tell your advisor everything was hunky-dory. You tell him the ambiguous data and why you think the conclusions are not as sound. It may mean that you have to go back and repeat some experiments. But repeating an experiment is a lot better than defending an incorrect conclusion. For an advisor, you might want to fudge things in your direction to help your chances for getting grant funding or for getting a publication, okay? These also apply to graduate students in terms of you guys will want publications and grant funding. Maybe you're going to go for some kind of fellowship in your first year. You could do a, say, an NSF graduate fellowship. You don't ever want to fudge data or make claims that are not clearly supported by the data you've generated. And then finally, promotions and advancements. You don't want to skew your data just because you know if you get good data, you're going to get a publication, you're going to get your Nature publication, and that is going to get you your promotion or your degree. You can't look at it like that. When you're in the lab and collecting your data, that's all you do is report what you see in a very clear and correct way. So when I was in graduate school, we had a postdoc from Germany, and I remember one day she came in, she was shaking her head, and I wish you could see me, but I'm moving my hands in frustration in front of my face now. And she was going, no, it doesn't agree. Why did we have to do this experiment? And then finally, after expressing her angst, she took a deep breath and said, well, these are the data, we cannot change them. And that was the correct approach. She took it into the PI, they discussed it. Yes, she had to do more experiments, but that was the worst that happened. She had to do some more experiments. They had to, after the new experiments, they reassessed their hypothesis. That is far, far better than having to defend shaky data. Trust me. So let's go through a couple of case studies. I think these are a little bit fun to do. So I've got 3 or 4 that we might go through today. And I'm sorry, you can read as well as I can, but I'm going to go ahead and read these out loud to you in case you've got screen fatigue. So, a principal investigator outlines her theory for a certain effect to her graduate student who is involved in acquiring data to either confirm or refute the theory. The PI explains her anticipation of finding experimental data that will support her, her hypothesis. The graduate student generates the data with approximately the anticipated values. Great, the data are published. The PI gets recognition. The PI gets her grant renewed. And the graduate student receives his PhD. Everything is good in that world. But the next student comes in and is going to continue the project. Okay, so the next student finds that he has difficulties reproducing the data from the first student. After further investigation, it's found that the first graduate student chose, whenever the data were ambiguous, he chose values that came closer to the PI's predicted values, thereby emphasizing a trend in the data that was not really present in general. So questions to consider. Should the PI have restrained herself from mentoring her or mentioning her anticipations? Should the PI have supervised the acquisition of the data more closely so that she could verify the accuracy? Should the PI have insisted on a more thorough check of data reproducibility? And what should be done now? Well, first of all, let's see. I don't have anything written about this, but I do want to talk about it. Should the PI have not mentioned what she anticipated the results for? There is absolutely nothing wrong with that. To send a graduate student in blindly to do a set of experiments without even telling them what the project is about, well, that grad student's being treated like perhaps a technician, and we're not here as technicians. I don't think we are. I think we're all graduate students or professional students. So you have to have a clear idea of the project, because when the data come out, you have to have a clear idea of whether the data support or refute the hypothesis. Even when we write grant applications, everything has to be hypothesis driven in this country, meaning you have to say, I think if we give this drug to these cells, we are going to have this type of response from the cells. We don't know that that's the response that's going to happen. In fact, we may have no idea when we write the grant application what will happen. Okay? But when we write the grant application, we have to make an assertion that we think this is going to happen. If it doesn't, we think these other things might happen. But if you write an application that says, well, always heard that turmeric was real good, so I'm just going to put it on the cells and see what happens. That's not going to be funded. That's not hypothesis driven. So you have to have a good idea of what you're doing. So yes, the PI should the PI should have mentioned her anticipations to the students, because the student is going to have anticipations anyway, whether or not the student understands the full project. The PI must supervise the design of the experiments and the process of acquiring, recording, examining, interpreting, and storing the data. Basically, the PI is responsible for everything. Okay. The fact that the first student was fudging data really does come down to the PI. Yes, the first student has some questions to answer as well. Okay. But the senior author on the paper is responsible for everything that's in the paper. So yes, the PI should have checked the notebook herself about the reproducibility. The PI should have at least once, if not more times, seen the student performing the experiment so that she could verify she had faith in the data that were being generated. And, you know, after you've been a PI for a while, you know when a student might have an opportunity to fudge data. We don't think anything's nefarious is going on. Okay, we just want to verify that we are teaching the correct scientific method to our students. We have a duty as scientists to avoid contaminating the literature with incorrect information. So the last question on the previous page was, what should the PI do about the situation? The PI should have the experiment run again, okay? And to see just how far off, because it's a new grad student, maybe the new grad student has a slightly different technique and hasn't developed his technique to the point that the first graduate student had. And so maybe the second graduate student can't establish those curves that are quite as high or quite as definitive. Okay. On the other hand, if she goes in and helps the second student, or the second student keeps repeating the experiment, getting consistent data that are different from what the first graduate student published, she needs to publish a correction. Ouch. That's a serious thing. Okay. So it's important that you as graduate students and laboratory workers generate good data, reproducible and believable, but it's also up to us principal investigators to verify and support that these are good data that we are presenting to the world when we publish. Case 2, outliers. This one comes up a lot. So if you're asleep, wake up. You need to listen to this one. Okay. So PI acquires data that portray a wonderful correlation between administration of a drug and a response in an animal. About 10% of the data, however, lie way far away from the predicted values. So we administer the drug, no drug, the control says, boom, we get 10 of whatever expressed. We administer the drug, boom, we get 80. And your error bars are really tight in there for the data that look good. But about 10% of the time, we get values around 10. Can we throw them out? Maybe, maybe not. And this is a key point, because you can actually throw data out sometimes, and that is not fudging. So if you can go back and look at your notebook, if you go back and look at the experiment and see that something happened, so, that, okay, hindsight's 20/20, but if you can note something that is not dependent on the data, that means that experiment is probably tainted, yes, you can throw those data out. They're unreliable. It's not that you're throwing out the bad values. If you had good values and the data were tainted, you can't count those either. So for example, suppose all the mice in one of your cages escaped from the cage and they ate some reagents in the laboratory that night. The next morning, student comes in, corrals the mice, and then collects data. In this case, yes, the corresponding data may be disregarded and left out of the publication. This counts for those outliers, but also remember, if you're throwing out data because those mice escaped the cage, you also have to throw away the data for the mice that presented the data you expected. Okay, so the moral is about the only reason you can ever throw out data is when something happened experimentally, or something happened that could have altered the results, and you didn't have to see the numbers in the data to determine, yeah, these could be thrown out. So like I said, that little laboratory mishap, that is an absolutely valid reason. However, if you say, oh, well, they're so far away, they count as outliers, I'm just throwing them out, and it's 10% of your data, oh my goodness, that's questionable. You're fudging at that point. We don't want to do that. Okay, the laboratory notebook. I'm sure you've all seen notebooks, but if you haven't, and by the way, when I started graduate school, I had never seen a laboratory notebook. You should see my first, no, you should not see my first laboratory notebook. I keep it as an example to my grad students of what not to do, okay? But I'm going to show you an example of what I show my grad students of what you should do. I'm going to tell you and then show you. They should be bound, your pages should be numbered, your entries should have dates on them, you should write in ink, not in pencil. If you cross something out, you cross it out to single line so that you can read what is behind the line, just to make sure you didn't go back after the fact and change some numbers. We all make mistakes, right? That's okay to make a mistake and correct it in the notebook. If you have an insert, you should sign it both on and off the insert. No erasures, no obliterations, and don't rip pages out of your notebook. If you screwed up on say a sketch, just put an X through the sketch and redo the sketch on the next page. Seeing an X through the sketch, if someone's auditing your notebook or your data for some reason, they'll understand why there's an X there. But if there's a page missing, they don't know what was on that page. Okay, so let me see. Here we go. Here I am. Lisa, wave if you could see me, the person. Okay, thank you. Here's my laboratory notebook, a laboratory notebook. First thing I'm going to show you is it is bound, okay? It's not a bunch of loose leaf papers that I throw in a 3-ring binder. This is bound with thread, and it is a fairly permanent document, okay? In about 50 years, maybe oxidation will do something to it, but it's fairly permanent. This is not one of those fancy notebooks that cost, boy, at least $29 each. They can go up to 50 bucks each, and they're beautiful, they're hardcover, they're bound. Some laboratories furnish those to the students, but if you have to furnish your own, I just use these Mead composition books. They're bound, they fit the bill, okay? My entries are all dated, okay? Yeah, this is old, but I'm not gonna show you the new notebook. But they're all dated. It's like a diary or a journal. I do have a couple of inserts here. You see, this is an image of a gel that I took with document camera. And let me find it for me first. Here's my name, and I have signed it both on and off of the insert. Okay. Yes, sometimes you need to take your inserts out and make copies of them. That's not as common these days in the digital world. But it's important that you sign on and off once it makes your notebook. That shows that you didn't 3 months down the road make a new image that looks better and paste it in for that day. What else do we have? Oh yeah, you can have qualitative data. I happen to just print out because it was so much easier for me when I talked to my PI about what the cells were doing. So I just took pictures of the cells and I pasted it in my notebook, and yes, it's still signed both on and off. So you can have qualitative data. You can have sketches. This is a preliminary sketch for an idea I had once. It actually turned into a patented device for seeding porous scaffolds with stem cells. Okay, but that's in my notebook. It is a permanent fixture, and a good thing, by the way, because when you get into patents, you have to show your documentation, the earliest work that you did on an invention. That date is very important. And I think there's one more. Oh my goodness, yes. Okay, yeah, you can load up with tons and tons and tons of data, but I actually marked this because I put the schedule for the entire experiment. This ton of data that I generated all came as a result of this experiment. So all of that is attached permanently in my notebook. Okay. Let's see. There are no erasures in here. Here's a good example. I'm gonna try to point to it. It says 3.48. I cross it and put 4.94. I think you can see that, I hope. But anyway, you can read the number behind it, and if you're really inquiring, you will see that this 4.98 is actually entered in the cell above it and crossed out. I put it in the wrong cell. It happens, but you document it that way. There's no question in my mind or anyone else's mind about what happened with that little mistake. So, with the Mead books, I have every page numbered. Yeah, I had to do it myself. This is just a hint, I'll tell you guys. I had this idea when I was a grad student of doing it, and it turns out the idea was a good one. Every page in every notebook I've ever written in, that's mine, has a unique page. When I fill this book, I start the next notebook on the next page. For instance, this notebook, the first page is page 341. And on the front, I'll tell you, in my career, these are pages 341 through 521 of my scientific laboratory career. That has helped me a lot. So just a suggestion for you, you don't have to do that, but you ought to have your pages numbered. I prefer to number pages all the time in my notebook. It makes it so much easier to find previous data that are included in the notebook. Okay, let me go back to sharing my screen. Here we go. These days, oh boy, let me try again. These days, we have electronic laboratory notebooks. Tulane has something called Lab Archives, and it's a cloud-based electronic notebook. It has a couple of advantages. One, it's free, okay, that's always an advantage. But two, it allows you to capture, manage, and store your information, but in a way that your collaborators can't. Oh, no, I'm sorry. Let me try again. In a way, oh boy. In a way that your collaborators can see it. You don't have to, if you're doing an experiment on the uptown campus, but your PI is on the downtown campus, you don't have to make a special trip downtown to show your notebook to your PI. The PI has access to it, and you could talk about it over the phone or via Zoom. So it's a great way of giving access to everybody in your team. Okay, and if you need more information, here we go. It's LibGuides or library guides, libguides.tulane.edu, and then just search for Lab Archives. Okay. Let's see. So I'm going to paraphrase this one for you because it's a lot of words. Grad student goes out, does work for the PI, works about 6 months. PI says, write it up. And by the way, let me see your notebook. Notebook does not exist. The advisor finds no notebook. Smith has been keeping records on loose pieces of paper. The records are undated and many cannot be found at all. What should be done and who is responsible? Well, the data are good. So we can go from there, right? Hopefully you're saying, no, that's silly. What can be done? I'm sorry, but if you didn't write it down, it didn't happen. In the world of science, if you didn't document it, it didn't happen. If you didn't put it in that lab notebook, it didn't happen. Shoving a bunch of I use paper towels, quite frankly, in the lab when I'm getting data and I'm in sort of a wet part of the lab and I don't want to smear my notebook and get chemicals all over it. I will jot down on a piece of paper, and for me, it'll usually be a paper towel. I'll jot down the readings as they come off the machine, but then I immediately transcribe them into my notebook. Okay? If you don't have them in a permanent manner inside your notebook, it didn't happen and you can't publish. Okay, who's responsible? Well, the grad student, yeah, but the PI, and probably mainly the PI, for not ensuring the student knew how to keep a lab notebook. Okay, so the PI has to pay for repeating the experiments. The experiments must be repeated, but it's the grad student who bears the brunt of the pain because it's the grad student that has to go back into the lab and generate all of the data correctly this time. So keep your lab notebooks correctly from the start. You'll never run into that problem. Data errors. We can have errors at the design level, experimental analysis level. We got a lot of different errors that I want to cover in the next 5 or 10 minutes. Okay, first of all, at the design level, maybe you have a sampling error, and that's when you don't sample enough. If I transfect cells once and they turn green and I'm happy, I can't really go home and publish on that one experiment. I have to repeat the experiment. And if I'm doing it with GFP, which I have several publications out on GFP, and if I'm showing that, yeah, I'm getting the same results as all these other published results, I can get by with 4, 5, 6 repeats. If I am testing a new drug on animals, I'm not going to get by with 3 repeats. Okay, you need more repeats. You can't do 1,000 with mice for several reasons. Okay, so you do have upper limits on how many repeats you can do, but if you don't do enough repeats, then the design of your whole experiment isn't really robust enough, and you're going to have trouble defending your data eventually. Your PI should teach you how to determine sample size correctly, and that may be by sending you to a statistics class. Okay, but you need to learn how to decide how big an experiment needs to be. I alluded to this earlier, skew or selection bias. So an obvious example would be to take a poll in front of the Democrat National Headquarters. Okay, I would expect to get a range of opinions that are different there than, say, at, well, in the middle of the Republican National Convention, which is going on now. So if I polled those 2 different groups, I would expect 2 different answers. If I wanted to represent the attitudes of the average American person, just taking this one poll is not going to do it. So the design of the experiment was faulty. I want to talk about skew too. This one's kind of interesting. Sometimes you could put skew or bias into your experiment without even knowing it. So I used to work with some extremely aggressive cancer cells. They're highly metastatic. And the fact that they were metastatic means that they didn't attach to their plates very strongly. So if any of you have worked with cells, you know that when you pass your cells, when they fill the entire bottom of your flask or culture vessel, you need to pass them into a new vessel at a lower seeding rate so they can continue to grow and divide. Well, with these metastatic cells, I found that I could pass them by tapping the flask on the side of the table, and most of them would detach. And then that would save me the trouble of rinsing the cells and putting them in trypsin and letting them sit. And if you know what I'm talking about, you know the rest of the story there. It's so much easier just to tap the flask, take 2 milliliters out, squirt it in a new flask. It saved a lot of time. And it seemed like a really good way to go until I realized I was selecting for the less adherent cells. Okay? At first, that wasn't a problem. But after several passages, all of my cells, 100% of them would come out when I tapped the flask, and that wasn't representative of the entire cell type that I was trying to describe. So I had inserted bias into my own experiment without even knowing it. So it doesn't have to be a psychological error that you made. It may be a procedural error. So you have to reassess what you're doing from time to time just to keep an eye out for selection bias or skew. Okay? At the experimental level, there's another type of bias where you disregard the results that don't agree with what you want them to be, or with fudging, and we already talked about fudging. At the analysis level, here's a non-intentional error you could make using the inappropriate statistical test. So if I have 3 treatments that I'm testing out, one versus the other, and I want to compare these 3 treatments to each other, a t-test is not the proper test. And I have rejected manuscripts for that very reason. They use a t-test, which is a pairwise test, and they use it to analyze 3 or more drugs. Okay. If you think about a t-test, it's associated with a 5% error. 95% confidence error means 5% of the time you're not confident. If you compare, if you do, let's see, well, 1, 2, 3, 4, 5, 6, all the different pairwise combinations of 3. Is that 6? 1, 2, 3. Okay, we'll do 3 tests, 3 t-tests. Well, that's 0.95 times 0.95 times 0.95. Give me a second. 0.95 raised to the third. We're at an 85.7% confidence level. That's not publishable. Okay, you need to do a proper test. And in this example, it's probably an ANOVA test, an analysis of variance test. Okay, so you need to be up on your statistical tests. Or maybe you forced your grouping. You know that the in a certain cell population, let's say the smaller cells are more easily transfected. So you just look at cells that are below a certain size. Well, that is a forced grouping. Okay, if the tissue or if the cells you're trying to talk about all come within a wide range of sizes, you need to test all ranges of those sizes. So you can make a data error in this way. Information bias. You got to make sure your machines are calculated. Of course, classify your subjects correctly. And then confounding error. This is another one of those from time to time you need to ask yourself. Go back and reassess what you're doing. Okay. Confounding error is something that you're not controlling for, but it's linked to both the independent and dependent variables of the study. Here are some examples. The incubator ran dry, and so the cells responded differently because of it. Mice were affected in your study because you didn't know it, but in the middle of the night, someone from, frankly, my lab came into the mouse room and turned on the lights for about 15 minutes so that we could administer a drug, and then we leave, and you happen to be doing a circadian study. This is something beyond your control that you didn't know about, but when you do find about it, well, at least you can address it. So if you start getting really odd results for no apparent reason, start getting creative. In my grad laboratory, if we couldn't figure out what it was, we'd say, oh, it's the phase of the moon. Okay, it probably wasn't the phase of the moon, but that was our code word for we need to try to find the source of this confounding error. Okay, you can have personal bias that leads to a data error. Those are the ones I talked about earlier. I need to get the graph to say such and such so that I can get the publication and get my PhD or get my grant funded. And then, of course, there's fraud, which is deliberate error. And I have this guy here because it can mean death to your career. Never, ever, for any circumstance, consider committing fraud in your data collection and reporting, because whether you're a grad student, a PI, or a Nobel laureate, that will end you right there if people find out you've been lying with your data. Okay, now, there are many different reasons why erroneous data may result in incorrect interpretations. Okay, those are costly. They have serious consequences. Maybe you have to print a retraction or a correction to the paper. We mentioned that earlier, but those are not fraud. Mistakes do happen. Okay, we had some. We were not as vigilant in monitoring our grad student. Whatever the reason, those happen. It doesn't mean we tried to fool everyone with lies. However, if you have a deliberate intent to deceive, you say you did experiments that you didn't, you say you did 10 experiments when you only did 3, you change the data so that it'll fit what you want the data to say, only count the results that agree with your hypothesis, just anything like that. Lies, don't do them. It's death to a career. It's instant death to a career. And then finally, this will only take a couple minutes because the answers are really quick. Who owns your data? Used to be, a long time ago, if you had an NIH grant or an NSF grant and you generated data and you generated an invention or something that was patentable, the government held the patent. The problem with that is fewer than 5% of that technology was licensed to industry, 5%. And so this is the Bayh-Dole Act, and it said a lot of things. You may have heard it within the past year. But one of the things of the Bayh-Dole Act was the inventions that come out, the patentable technology, the university can hold the patent, not the government. And now more great ideas and discoveries are being patented and marketed, and there are more startups. And that's one of the reasons we have such a flourishing technological world in this country. There are 3 things I want to make you aware of in terms of regulations in Tulane. There is the IRB, well, I can't, sorry, trying to highlight things. There's the IRB, the Institutional Review Board. If you're going to do anything with humans, you have to have the IRB involved. If you're going to do anything with mice, the IACUC must get involved, Institutional Animal Care and Use Committee. Okay, and if you have any general questions about compliance and research integrity, here is the link. Notice all of these are research.tulane.edu. That has been more and better formalized since the last time I gave this talk. I'm very happy to see this organization. So it's easy to find these. You don't even need to remember these last extensions. You can find them all through research.tulane.edu. Who owns the research that you do at Tulane? Tulane owns it. Okay, even though you probably will have some great ideas when you're doing the experiments, or you suggest an experiment that to your PI that you want to try for your project, and the experiment yields and shows that an idea you had is valid, you can't run away from Tulane and say, I own that, I own the patent on that. You can't go patent it on yourself. Tulane will be happy to help you. And I will also say Tulane has a beautiful and generous patent policy, okay, compared to other universities. The Intellectual Property Office says disclose, and disclose often. Do it. It's in everyone's best interest. Okay, it'll be your PI who manages the data. Tulane will own the patent, but if the principal investigator wants to form a company around the patent, Tulane is also very generous in making that happen. So keep Tulane involved, not just because you have to, but because they're a great ally to have on your side. And I can speak from experience on that. Okay, rather than go over this last ownership thing, it talks about what I just told you. The only other caveat I want to say, and most people don't know this, if a graduate student comes with an idea and proves that it's good, the grad student can't patent it on his own. However, if an undergrad does it, the undergraduate can. The difference is you grad students and professional students, well, you grad students are technically employees of the university. You use the university's equipment and reagents to get the data to support your idea. So of course, Tulane's involved. An undergraduate is not an employee of the university. In some eyes, the university is an employee of the undergraduate. So things get a little different. But if there are any undergraduates in the audience, let me tell you, don't be so secret about your idea that you don't tell anyone and think you're going to develop it on your own after you graduate. Tulane is a great ally, and the advice that they can give you for developing your idea into something patentable and licensable, it is absolutely worth bringing them in on it. And with that, I am through with the talk. I will be happy to take any questions if you have any, and I'll give it back to you, Lisa.
Dr. Godby, thank you so much. That was fantastic. We really, really appreciate you presenting today. Does anyone have questions? You can type them in the chat. Or Tim, I don't know if you want to unmute everyone. They can ask a question verbally.
They can go ahead and unmute themselves. They're ready to go.
Okay, so if you have a question, you can unmute yourself. While we're waiting to see if we have a question, I'll just note next week's class will not be held on Zoom. Next week's class is the only class that will be held on the Teams platform. You will receive an email invitation from the presenter for the Teams link. Please accept that invitation, and that is the link you will need to join next week's class, and that's only for next week's class. After that, we'll be back to Zoom. Does anyone have any questions for Dr. Godby?
Hi, I have a question.
Yes.
So I loved seeing your notebook. I had one in at my last job, and I was just wondering. I was taught that you could, that I needed to, if I made a mistake, I needed to cross what I did wrong, and
Yes.
then I would have to write above the mistake my initials and my signature and the date that I did the mistake. For example, is that like a regular thing, or is it okay just to cross and right above the mistake?
It really depends on the institution that you're at. You were at a company, you say?
Yes, yes. Mhm.
Okay. So anytime money's involved, things get a lot more serious. So where I showed you, where was it? It was one of these. I showed you these, I crossed it out, I put the new number in. I think it's entirely reasonable to ask you to initial that, okay?
Oh, okay.
Because it shows that you made the correction. In my notebook, it's pretty easy to tell I did it. I had bad handwriting. But I could see why a company would want you to initial it. The other things that you said are probably company policy. They're just covering themselves from a legal standpoint in
Okay.
case they ever get challenged, because frankly, there's going to be a lot more money involved.
Okay. Okay. And I just had another question. It was about data ownership. For example, if I'm working at a trial and we're working with humans, so we needed to sign informed consent. So
Yes.
we got all the signatures and everything, and now we have the data.
Yes.
But let's say 10 years from now, we want to do a different kind of study with that data that we already have.
Mm.
It wasn't like the initial study. It is a different study with that same data. Am I able to, or the company, for example, is able to use that data for a different study? Or am I not allowed to use it since the informed consent did not mention anything about it?
Okay, we'll talk about it in terms of your company. So when you did the first study, the company owned the data and the company maybe published the results. They did whatever they did with the result. Now, let's say you're a grad student, you would like to analyze those data in a different way. Even though you helped to generate the data, you don't own those data. But you can write the company and say, hey, I'd like to do such and such study. Would you allow me access to the database for that? Some companies will say, oh, Anna, I know you, and this sounds good. Yes, go right ahead. The next step would be they'd say, well, yeah, Anna, I think that'd be interesting. We want to be a part of that study, meaning we want access to what you figure out, and we want to be on the paper. Okay? So you either acknowledge them in the paper, or there might be someone you have to include as an author. And then other companies might say, oh, well, we can grant you access, but it's going to cost you this much money. So every company has its own philosophy. Every university has its own philosophy. So the bottom line is you just have to go to whoever owns the data and ask them. And they're not as mean as you might be thinking. It's usually pretty nice. But especially if you consider the alternative is going in and, you know, involving another 100 or 1,000 people. If we can get more conclusions out of that initial set of questioning or experiments that you did, that's just great for humanity. And most owners of data will recognize that and be happy to work with you.
Oh, okay, thank you very much.
Sure.
Other questions before we wrap up today?
Hey, Dr. Godby, I had a question. So most of my work is computational. Use a digital lab notebook, but I write in plain text markdown. I kind of shy away from proprietary oriented lab notebook things. What would be your suggestion as far as, let's say, for IP purposes, making sure you confidently record the date and times and data in a way that an outsider would be able to verify it? The closest thing I can think of is some type of version control for plain text, but if you have any suggestions.
Okay, so first of all, I want to ask you, are you a graduate student or a PI?
I'm a graduate student.
Okay, so the answer for you is you should go to your PI and ask, because when we write to different granting agencies, they have different rules on how we store our data, store and manage the data. And so it's not for you to know. It's not a secret from you. I'm just saying there are a lot of different ways, and the different agencies have their own rules. So your PI will know the rules and whether you can keep doing it in plain text. If you are allowed to do plain text, version control is probably the easiest and most straightforward way to do it.
Gotcha.
Hi, Dr. Godby. I had a question about lab notebooks. This is
Yes.
Caitlin Montgomery. So I saw that you were showing your lab notebooks, and so I wanted to know who owns your lab notebooks. Is it the graduate student? Is it the PI? Is it the school? Are you allowed to make copies to keep with you throughout your career? Do you have the answers to those questions?
Yes, I do. And that is a very good question. I thought if I paid the money for these pieces of paper, I owned these pieces of paper. I paid for the pens that had the ink that wrote on here, so I owned the ink that was in here. And while that may be true, technically, no. This becomes a piece of an extension of intellectual property, and it's documentation of work that was done. So, well, we'll say this was at Tulane. Work that was done at Tulane that Tulane owns. So Tulane actually owns this book. Am I allowed to make a copy? Yes. Do I? Well, I've got the original. But if this were my PI's notebook, meaning he owned it because of the intellectual property, you better believe I'll make a copy. In fact, all of you need a copy of your notebooks from time to time, copy, like every month. Copy everything you did for the month and store it off site. For anyone from around here, 2 words: Hurricane Katrina. For the rest of you, what if your laboratory burned down and all of your documentation is gone? Keep an off-site copy and do it for your whole career. Don't do it in secret. Let your PI know, okay, I'm making a copy, I'm keeping it in my apartment, or I send it in my grandmother's house, whatever. You know, whoever actually owns the original should know, but they will have, I guarantee you, your PI will have no problem with you having your own copy as a backup. Does that answer all the questions you asked, Caitlin?
Yes. So if you, for instance, my PI moved out of Tulane and took the notebooks with him, I made a copy for myself, and so that's why I was asking. Thank you.
Yeah. Oh, good. Well, that's a sticky one. So if I were to leave Tulane, and I've got many notebooks that were generated at Tulane, I would talk to Tulane and say, who gets custody of the children here? I can expect that no matter what they say, whether I keep the original or they keep it, a copy will be made. Okay, and either I'll leave with the copy. I wouldn't leave. I'm not leaving. But if I left, I would either leave with the original or the copy, and the other one would be in possession of Tulane.
Any other questions? All right, Dr. Godby, thank you so very, very much for presenting today. We very much appreciate it. And we will see everyone next week on Teams. Be on the lookout for the email invitation. Thanks, everyone. See you next week.
Thank you.
Thank you.